It's another edition of the Air Center Networking Fridays. Today, we are discussing an important topic, and because there are a lot of challenges affecting ocean, particularly from land, due to poor waste management. However, in some developing part of the world where most water bodies are channeled into the ocean, there are spread of various marine litters into the ocean. And in Nigeria, a scientist and a researcher with his colleague in the department and in the research group are working on some preventive approach to Atlantic Ocean and other environmental pollution in order to achieve a circular economy and sustainable resource recovery. Permit me to introduce Dr. Buraimoh Maria. Before that, I would like to welcome everyone to this program again, particularly our distinguished audience. I have the information that today we about more than 100 people registered for the program from 31 countries. And it's important to note that more than 50% of attendees on this platform every Friday are from Africa. And just let me to share this also that about 2/3 are from Nigeria. So everyone, you're welcome to this event. And a brief citation about Dr. Olanike Maria Buraimoh. She's a lecturer and researcher at the Department of Microbiology, University of Lagos. She has a PhD at the University of Lagos, and her research interest includes environmental microbiology and biotechnology, with key interest in bioremediation of polluted ecosystems and bioconversion of waste to value-added products in order to put them into sustainable solution and prevent climate change. She has a lot of awards. She was a postdoc candidate at Karlsruhe Institute of Technology here in Germany, and also a visiting scholar at Ohio Agricultural Research Development Center in the United States. And she was the project event manager, Science Ambassadors Foundation. Some awards to his credits include the Faculty Best Researchers Award, Tertiary Education Trust Fund Research Grant, then TIWAS DFG Corporation Visit Grant Award, then International Center for Genetic Engineering and Biotech Training Grant. She was part of the inaugural program workshop of the Air Center in Nigeria at the National Space Research and Development Agency in Abuja, and was part of the Atlantic Interaction 5th High-Level Industry, Science, and Governmental Dialogue in Nigeria. So she is an associate of the Association of Massachusetts Institute of Technology, Train African University, a fellow of the Institute of Policy Management Development, IPMD, and a member of the Society for Environmental Toxicology and Pollution Mitigation, SETPOM. And is affiliated to so many organizations. So personally, my name is Elegbede. I am affiliated with the Brandenburg University of Technology in Germany, and my research area includes certification, marine resource management, and circular economy, blue economy, fisheries, and certifications. So we both have some things in common. We are both alumni of the University of Lagos, and we also passed through Germany as well. And so that's just brief about our discussion. So I would like to give some notes dear audience, please kindly use the question and answer portion of the platform, of the Zoom platform, and it's better to use the Q&A portion so that we can easily read some of your questions. So on this note, I will invite our distinguished doctor, because today I was told by Jose that Maria has been a popular name about some generations back in Portugal, because most people usually have Maria with their names in Portugal. So on this note, Dr. Buraimoh, you have the floor. So you can now share your screen now.
All right. Thank you very much, Isa. Thank you for the introduction. Good afternoon from Nigeria, but I say good morning, good afternoon from wherever you are networking from. So I'll just start my presentation immediately without delay. And so I'll talk about circular economy, a sustainable and preventive strategy for the alleviation of Atlantic Ocean pollution. So I will be guided by this outline here, starting with the introduction till the last. So what is the concept and theory behind circular economy? It's all about sustainable development, green economy, performance economy, industry ecology, extended producer responsibility. It's about eco-friendliness, life cycle, cradle to cradle, not cradle to grave. It's about innovation and share value that has to do with solving societal challenges. Now, economy, usually what we have around this, around Nigeria is what is called linear economy, a situation whereby you take a natural resource, you make them into products, you make use of them or you consume them, and then you discard. But in circular economy, we are talking about taking a natural resource, making it into products, making use of the products, recover it, remake it into another usable product, you reuse it, then you circulate it back into the environment without causing harm into the environment. So in essence, that is what circular economy is talking about. The circular economy system, it's in two diverse forms, two systems, the technical and the biological. And as you can see here, because I'm a microbiologist, I'm dealing mostly with the biological economy system. Not as if we are not delving, gradually we are delving into the technical aspects. So what we are talking about is using biological means that is of eco-friendly to resolve waste in the environment and converting it into value-added products. So what are the major challenges of the ocean in Nigeria? The major challenge we have is that of poor waste disposal, poor waste management, and other human activities. Waste generation and management in Nigeria has become a big challenge because of the increased population, the issue of urbanization and industrialization. In Nigeria, more than 32 million tons of solid waste is produced annually, and out of which only 20 to 30% is collected. Out of this 20 to 30% that is being collected, 15 to 30% is made up of plastics. This is worsened because we have very poor storage facilities in Nigeria. Now, Lagos State is a good model in Nigeria. Lagos State is one of the biggest cities in Africa, and it is the commercial hub of Nigeria. It has a population of over 14 million. Some literatures says 15 to 21 million. But Lagos generates 9 to 15 tons of urban waste, and 30% of recyclable waste. 50% of this waste is also plastic. Lagos State is one of the states that is at the forefront of municipal waste management. They've done a lot. They are making move, they are already making move to generate billions of naira from waste recycling. Ojewale 2019 opined that if 500,000 tons of plastic is recycled annually in Lagos at the rate of $500 per ton, it will potentially be a 250 million industry, and a significant increase is expected when other parts of Nigeria are factored in. So it's a big business to manage waste. This is a typical waste disposal system that we have in Nigeria. This is still what we have typically. Popular dump sites in Lagos, this is what it looked like, with all sorts of waste being mixed together. We have situations where the waste are burned, they are set on fire, and then you have a lot of gases being released, the greenhouse gases being released into the atmosphere, further degrading the atmosphere and then leading to respiratory challenges in humans and animals. We also have these typical situations where we dump waste into the canals and drainages, especially during the rainy season. So this will either block the canal or get washed into the ocean. We also have situations where we have shanties and houses are built on top of the lagoon. Here, they do not have special system for toilets. They defecate and do everything they want to do on top of the water. They live practically on water. Their life is on water. And so we can see from this graph that the rivers, the lagoon, the creeks are all connected together and ends up in the Atlantic Ocean. So typically, what we find in the ocean, the kind of waste we find in the ocean include the metals, the lignocellulose materials, the wood residues. We have a lot of plastics, some organic waste, and so on and so forth that are being dumped into the ocean. So what I'm trying to say is you have sewage, industrial effluents. We all know that in Lagos, we have several oil facilities and depots, and their effluents are connected to the ocean because actually the vessels bring the petroleum products from other parts of the world and they are discharged. So we do have a lot of spillages. And then you have other human activities, all ends up in the lagoon. You have the plastics, the hydrocarbon, the lignocellulose materials, and other organic materials ending in the Atlantic Ocean. So, in essence, the ocean has become a repository for waste disposal. And invariably, waste generated here in Nigeria move to other continents of the world that have shared ocean space. So, the processes, when waste are coming from the homes, from the offices, industry, restaurants, and so on, it goes straight to the dump sites, and then the seepage, the wind, erosion, and flood can take it straight into the ocean or transfer it to the drainage, the canals, the streams, the rivers, and the lagoon, and then they are channeled straight into the ocean. And then we have situations whereby there are direct dumping of waste into the ocean. So we can see that the ocean is grossly polluted in this part of the world. What are the ecological effects of these residues on water bodies and the ocean specifically? One of the most important one is the global warming. When you have water pollution from maybe organic materials, it leads to addition of nutrients. If you have them in form of plastic or microplastics, it can affect the fishes. We all know of this whale that swallowed about 68 or 69 grams of plastics, and the whale died, and then They discovered a lot of plastic, and fishes have microplastic in them, and these are dangerous when we consume such fishes. So for addition of nutrients, organic material or lignocellulose material decomposes to release aromatic compounds into the water. This causes additional nutrients in the water body and leads to algal bloom and other water plants eruption. This intensifies sea ice decline, and as the Antarctic warms up, more sunlight penetrates the ocean. This triggers more growth in the water plants, and they absorb an increased amount of sun's energy, which causes the water to warm up. And a warmer ocean means more sea ice melting, which invariably leads to global warming. Other problems has to do with enhancement of microbial growth. Of course, when you have additional nutrients in water, it leads to microbial growth, and it leads to disruption of the ecological balance. You have increase in oxygen demand, clogging of waterways by these water plants, occlusion of light, toxicity problem for polyaromatic hydrocarbons, death and migration of fish and other aquatic organisms. Then it is noted that eruption and spread of infection from further ocean warming is prominent. So we have to be careful what we do to the ocean. So what is the way forward? Myself and my colleagues, we have tried to look at what can we do as microbiologists, as zoologists, to minimize the contamination or pollution of the ocean. So in our own little way, we thought that prevention is always better. It is better to be proactive, to apply proactive approach rather than packing plastics from the ocean or trying to pack waste from the ocean. It is better to prevent the waste from getting into the ocean. So how do we go about this? Through proper waste disposal. There has to be a lot of education to educate the populace on how best to dispose waste, through proper collection, sorting, and management. Then recycling of waste. Moreover, I am more interested in the bioconversion of waste to value-added products using physical, mechanical, or biological means. And the biological means, we are talking about using indigenous microorganisms that are not harmful to the environment. So this is usually done with the deliberate intention to prevent pollution of the ecosystem, while at the same time generating incomes and jobs, because we have a lot of poverty syndrome in this part of the world. There are no jobs. So by doing this, you create a lot of jobs and add some flesh into the economy. And some of the products that we can obtain from this is clean energy, which includes the biogas, the bioethanol, the biochemicals, bioenzyme, and biofertilizer, and so on. The list is inexhaustive. So 2030 sustainable development is almost gone, because 2030 is almost here. But remarkable progress is still very slow in Nigeria. And it is amazing to see that majority of the UN SDGs contain targets with a direct bearing on waste and waste management. For example, SDG 1, no poverty. So if we are able to create jobs from waste management, it will reduce poverty. Good health and well-being, number 3. We are talking about management of ecosystems. So when the ecosystem is clean, the health and the well-being of the populace will be guaranteed. Quality education. We need quality education to educate the populace. People that are not educated cannot understand what we are talking about, so they don't see the reason why they should not dump their waste in the ocean. You know, we are talking about clean water and sanitation, affordable land and clean energy. When you bioconvert to, biogas and others, you are talking about clean energy, decent work and economic growth, because circular economy leads to economic growth, industrial innovation and infrastructure, and all others, 11, 13, 14, and all these SDGs are connected to waste management. Transition to circular economy. If we look at this diagram, you can see that these countries are represented: Scotland, Brazil, the US, the Wales, the UK, the India, the EU, they've done the China, Japanese, the South Korea, they've done a lot of work about circular economy. But where is Nigeria here? We can't find Nigeria except for Lagos that is making move, and it is beyond Lagos. So we have to act fast. We need to do something. And not that we have to work together even with other countries that have shared boundaries with Nigeria concerning the ocean. Because as we can see, whatever comes out of Nigeria as waste into the ocean will also can flow to any part of the world. So what do we require to transit to circular economy? We need multidimensional value assessment tools, which integrate the environment, the social well-being, and the economic factor. In short, collaboration and participation of all stakeholders is very essential, as no single person can do it alone when it comes to circular economy. This is what I'm talking about. Belentov and Cornell 2017 have a guide. They have this circle that shows that to tackle this problem and for you to have a proper circular economy, you must form a stakeholder group. You must identify what the problem is in your own area and try to find solution. You discuss, you share ideas, then you evaluate your process. There must be societal change. If there's no change, the process has to be repeated. So transiting towards a circular economy must be encompassing. It must involve all the stakeholders, from the designer of products to the manufacturers, to the consumers, the waste processors, the academia, the policymakers or regulator, and then the NGO. For example, evidence-based supporting technologies and analytical tools are provided by the academia. This is not the time for academia to perform experiments and keep it on the shelf or simply just publish it and leave it there. This is the time for the academia to come out of their shell so that there will be synergy between the industry and the academia. The policymakers need to provide legislation and enforcement and support, because sometimes the citizenry will not just obey some of these rules if they are not enforced by the government. And of course, the NGO can provide support where the government fails, because governments cannot do everything on their own. And designers may redesign products to use a single polymer, for example, in plastics. Either you use a single polymer that are standard that can stand the test of time, or design it in such a way that different materials can easily be separated. This will aid recycling. So what are our actions so far? After laying this foundation about circular economy, I'm a microbiologist, but I have delved into this area because I find interest in it. How did it all started? It started during my PhD work. My supervisor asked me to isolate some microorganisms from a Ebute region. In this place, we have a lot of sawmills, and a lot of residues of sawdust are dumped into the lagoon. As you can see, the bridge, the Third Milan Bridge, and under, directly in the water, they dumped their sawdust and other residues there. And methane, you know, is generated after a while. Sometimes they also set it on fire. So I thought about it that the best way is to isolate my microorganism from here. Microorganisms that are already used to this sawdust definitely will be able to take care of the sawdust. Because take a look at it. This sawdust, these sawmills have been here year in, year out, and they kept dumping these materials in this water. By now, the lagoon should have been filled up and should have broken down. But definitely, I'm sure it is because of the microorganisms that are here that are helping to regulate the system. So I collected my isolates from here. And then I collected water samples to do the physicochemical analysis of the studied site, so that I will know what kind of condition to apply to the microorganisms in the lab. So this is the satellite image of the Lagos Lagoon showing the location of sample collection and the in-situ studies. So I was able to isolate, at the end of the day, I isolated several organisms, but those that showed technological competence were about 9. But the most interesting thing is, while doing this work, I was not having a circular economy in my head. All we were after is to get microorganisms that will be able to digest the waste residues. So we're able to isolate very good strains. And then I was able to also do the physical chemical parameter of the location. And it shows that the copper, the lead, the zinc, they are so high compared to Nigeria local standard and the WHO standard. So showing that the lagoon is contaminated and the lagoon is channeled into the ocean. Again, I tried to look at it. Definitely, I was unable to isolate all the organisms in that environment. So we tried to look at the microbial diversity of decaying wood since we isolated from decaying wood. So using the terminal restriction fragment length polymorphism and the Illumina sequencing, we were able to look at all the microbial diversity that are present in the decaying wood. And the result shows that 72.56% of the organisms inside the decaying wood we could not isolate. So there are a lot of unknown microorganisms that are working to degrade the wood and are working to help us to stabilize the ecosystem. So the actinobacteria and the firmicutes that I was able to isolate were just 6.4% and 1.36% within the decaying wood. So the microorganisms of this marine area, this lagoon, are very, very important. They influence our climate. They play a major primary producer role in the ocean or in any water bodies they find themselves. They dictate much of the flow of marine energy and nutrients. They are also the sources of medicine and natural products. We know of corals, we know of several plants that we can get from the ocean, from the water body that are of medicinal importance. Moreover, the reason I chose these marine isolates was because they are already adapted to the harsh environmental condition of the marine ecosystem, which gives them that advantage of technological competence. So we started work after isolation. So we're able to deduce that if we must use these microorganisms for bioconversion or to degrade organic matter or the wood residues, they must be able to remove craft lignin. Craft lignin is a typical lignin substance or material. We know that plant material or lignocellulose materials are made up of coniferyl, synapyl, coumaryl. They are made up of lignin, hemicellulose, cellulose, and the lignin is the one surrounding it. It is very recalcitrant. So if the organism is not capable of at least removing a portion of the lignin, it will not be able to penetrate to utilize the cellulose and the hemicellulose. So we subjected these microorganisms to fermentation using craft lignin as the substrate. And from the table, we can see that they have varied ability to break down craft lignin. But it is amazing when you see Bacillus batavensis here, it was able to degrade up to 90% of the craft lignin with a specific growth rate of 0.90 and doubling time of 9.12. But one should not be carried away with this result because most of the organisms that were able to remove craft lignin could not make use of cellulose and the hemicellulose. So probably they removed the craft lignin so that other organisms will be able to penetrate to make use of the cellulose. So the Streptomyces arborescens and Streptomyces silicolor that did not do too well with craft lignin actually performed better and this organism together with Bacillus megatherium were taken for further studies. Up till now, we are still using them. So we also checked for the ability to degrade lignin-related aromatic compound. It is known in literature that fossils are formed as a result of accumulation of plant material over billions of years. So meaning that if fossils and hydrocarbons are formed from plant material, then our organism should be able to degrade lignin-related compounds, which are components of plant material. So we checked several of lignin-related compounds, and we can see that these organisms have varied ability to degrade the lignin-related compounds. So from there, we're able to see what is the metabolic product of all the degradations that we have done. What can we have there? Because most of the microorganisms are bacillus or streptomyces, and they are known to produce metabolic products. So we did extraction of the culture supernatant and then subjected them to HPLC, MS, and UV spectrophotometry. We also carried out sequential growth on the metabolite, and were able to find or to be able to project the pathway of some of these microorganisms when you feed them on certain substrate. So this is a table showing the identified metabolic products. And with this, we're able to put together the metabolic pathway for this specific organism using coniferyl alcohol alone. We did several. I'm just showing us a bit of the example. But something amazing happened. And in between, during the construction of the metabolic pathway, we discovered the presence of cafeolpinic acid and coumarylpinic acid. The two acids are a group of chlorogenic acids that are of medicinal importance or relevance. They are known to cure several diseases. They are part of several drugs that are used to cure diabetes, high blood pressures, and some cancer. So now we know that if we are able to have plant waste that contains a lot of coniferyl alcohol, because different plants have different quantity of these components. So if you are able to have plant materials that have high percentage of coniferyl alcohol, you can play around with some of these organisms and have your chlorogenic acids, which are of medicinal relevance. So we went further to see, okay, so that means these organisms have these enzymes that could break down lignin, that could break down cellulose and others. So we have to carry out the enzyme activity. So we did several enzyme assays. So I'm just showing you two of the assays. Like the first one is peroxidase activities, the cellulase activity. We did all that for the project, but now it's making more sense because this is an indication that I can actually use waste as a substrate and subject my microorganisms, allow them to grow, to release the enzymes into the medium, and then I can harvest my enzyme either from the medium, or if it's an intracellular enzyme, I can obtain them from my microorganism by breaking them down and collecting the enzymes. And we know that enzymes are very useful in the industry, be it medical industry, pharmaceutical, food, or whatever. So we try to look at some of the organisms that performed very well. Do they have these gene clusters for these enzymes in them? So we were able to use some metagenomic devices to check if they are actually carrying the gene clusters for some of these enzymes. This specific table is for cellulase enzyme. So we have been able to deduce the kind of gene clusters that they have in them. We also did for dimethylase and protocatechupic acids. So from there, we proceeded to in situ studies and laboratory studies on the wood residue. I haven't established that the microorganisms have been able to degrade the components of the wood residues, and we've been able to isolate the enzymes that are supposed to be used, and we've been able to see the various products from them. So we proceeded to the practical aspect, the in situ study. So we carried out the in situ study, making use of the floating raft equipment that we made from the styrofoam. as we can see in the diagram on the left. And then we were able to use some certain grams of the sawdust and inoculated them with the microorganism. We tied the surface with a 0.2 micron filter, and then we turned it upside down and kept it in the lagoon for 6 months to see what will happen to them. At the same time, we also carried out laboratory studies for applying the same method, the same quantity of wood residue. So at the end of the day, we discovered that the in situ degradation study using the isolate was better than the laboratory studies. We know there are several reasons, but which I cannot start stating here. So for example, the Streptomyces AOB was able to remove 58.7% of lignin, whereas in the laboratory studies, only 47.5% of the lignin was removed. If we look at Bacillus megatherium species also, for the in situ study, it was able to degrade 82.52% of lignin, that is natural lignin, and then for the laboratory studies, 52.49% of the lignin was removed. So I haven't known that, of course, the PhD is over and all that, and I was back to myself. And during my work in the US, I discovered something. I discovered that during the degradation of the sawdust, I came in one afternoon and I started smelling ethanol. I thought, oh, maybe because I was tired and I left the bottle of ethanol open or something. But after a while, I discovered everything was intact. So I was able to trace the odor to the shaker. And so this odor was coming from my experiment. So I called my supervisor. I think these organisms are producing bioethanol or some other products. But my supervisor said, face your degradation. I didn't send you there to go and do bioethanol work. But then I had it at the back of my head that after the whole PhD, I was going to go back to that work. I want to see what the microorganisms are producing. I also checked the literature, and I was able to see that different waste can give you different energies, can give you different material, ranging from antibiotics to antioxidants and all that. So here on this table, we could see different plastic materials, and we could see the amount of energy they can actually release. And the municipal solid waste also, the newspaper, the textiles, the wood, which I was actually working on, and the wood waste. So I decided in my mind, I was going to start working on each and every one of them to see what I can actually obtain from there. So that was when the issue of circular economy started ringing in my head. So here, this work was done in Germany, and I took some plant residues from Nigeria and these organisms with me, and I subjected them to degradation or fermentation. Here on the left contains a specific microorganism, the one in bead, that was grown using pure glucose, and the brown one was grown using a particular plant residue. As we can see, both of them are comparable. The pure glucose growth is not any better than the growth obtained from the waste. So, and glucose is very expensive. Moreover, in Nigeria, we spend money importing these things for laboratory use and industrial use. So using waste, we can use waste as substrate to grow microorganisms, either for biomass production, as we can see, as we have here. Some of these biomass can be used as additional nutrients for feed, and then, or using waste to grow the organism just to obtain the enzymes. For example, if I want to obtain my enzymes from these two isolates, I can collect the enzymes that have been released extracellularly. I can also break the beads of microorganisms and collect the intracellular enzymes. So these are the products. On the right-hand side in the bottle, we have different biochemicals. This work is already patented. We have different biochemicals of industrial relevance in here. And the brown one was made using waste and microorganisms. The left one was made using pure glucose. And from the analysis, the results of the microorganism fermentation, the fermentation using waste was better than the one using pure glucose. So in 2016, I met a very interesting woman, Wilkie, from the University of Florida. And she got very interested in the work I presented. So she told me that Africa is still behind when it comes to circular economy. She showed me some work which she has done. I got this diagram from her. So I made up my mind I was going to, I'm not an engineer, but I have engineers around me, and together we could put our heads together to make some of these things work. So we started gradually by collecting bio waste such as discarded fruits, discarded leaves, and so on, and to convert it to biogas. Where we are actually going is a kind of combined heat power unit, where we'll be able to generate electricity, heat, and biofertilizer from waste. So we started actually, and then myself and my students, and Professor Ogundipe really encouraged me, the VC of the institution, because he's overwhelmed by the level of waste production on campus. So he needed a situation where we'll have something to do with the waste, to get rid of the waste. So he was really very, very interested in the work. So we tried the food waste around the campus, and then we subjected them to anaerobic digestion, and we used cow dung, cow parts, as a source of microbial inoculum. So this is the diagram before the production of gas. As you can see, the tires are all flat. And this is the diagram after biogas started producing. Initially, the gas was not pure because it was orange in color, but after about 5 days to 7 days, we began to have eco-friendly biogas. Now, I discussed this discovery with some engineer at in a non-governmental research institute. They have a lot of funds to play around with, so the whole thing was very fast. And within 4 to 6 months, they already have a very high quality biogas that has a very high, now they've even gone higher. The tank is very massive now, and the canteen is actually using biogas for cooking. They no longer spend money buying gas. So we proceeded. Plastic is a major problem in the ocean. How can we deal with plastics? So we thought about it. Plastic resins are actually made from fossil fuel. So if hydrocarbon and fossils as a result of the degradation or the decomposition of leaves and plants over billions of years. So meaning that this wood digesting microorganism should be able to degrade the hydrocarbon and invariably should be able to work on the plastics. So we carried out some experiments to check the efficacy of intervention strategy for bioremediation of crude oil in polluted soil microcosm. This work has been published since 2017. What we try to do is to deliberately contaminate the soil by adding crude oil to it. But intervention strategy there means that for some of the isolates, we added nutrients like the potassium phosphate and the sodium nitrate to it. So from the bar chart, we could see that the microorganisms also have varied ability to remove the hydrocarbon from the soil. But the organisms that has nutrients performed better. We went ahead to see the potential of hydrocarbon utilization by this wood degrading microorganism. And we can see that the Bacillus megatherium was able to remove the hydrocarbon from 3,481 milligram per liter. And after 21 days, the remaining hydrocarbon was 371 milligram per liter. The same thing goes for other organisms. So with that result, we decided, I gave one of my MSc students the work to do. Let's start with polystyrene plastics. So we're able to treat it mechanically, and then we subjected it to fermentation using only the plastic, no nutrients, no heating, so that we will not further, degrade the environment. After about 5 weeks, we subjected it to GCMS, and it is amazing that the GCMS results compared with the control shows that some components, toxic constituents of the plastics, have been taken up as substrate by the microorganisms. So oxalate, dodecanoic acid, and cis-hexadecanoic acid has been removed. They are waste, they are toxic wastes that are dangerous for the health of humans, and the microorganisms have been able to take them up. Other selected work, we've also tried to do several things. The spent from the anaerobic digestion, we try to use as biofertilizer. So another master student worked on it using selected vegetables. So we got some very good results. We've already submitted the work for publication, and it is under review. I worked with my colleague who is a pharmaceutical microbiologist. So I gave him some of the isolates, especially the streptomyces, that he should check if they are producing some antibiotics. And we got some amazing results. Some antimicrobial activities were found in some of these streptomyces species, and the work has been published since 2017. We try to see, because pectinase and some other enzymes are very useful, especially in winemaking. The cellulose is using in textile, is very useful in the textile industry for the removal of stain and all that. How can we obtain enzymes from this same isolate? We started with pectinase, and we saw that the bacillus species were able to produce pectinase. The work has been published since 2017. I also carried out some experiments with my colleague in the zoology department. So we were able to look at the Institute toxicological evaluation of effluent from a petroleum depot on hermit crab, and then the bioremediation using indigenous microorganisms. We had some very interesting results, and we made the presentation at the 12th University of Lagos Annual Research and Fair, and the paper has been prepared and also about to be submitted for publication. Then I considered that in Brazil and in the US, okay, the Brazil makes use of sugarcane a lot for the production of bioethanol, and they use this bioethanol, as gas for running their automobiles most of the time. So since I want to take care of the waste, we have a lot of sugarcane baggers waste. So I decided to see, using 2 of my isolates, to see if they will be able to take up the sugarcane baggers as substrate to produce bioethanol. And we have very amazing results. I presented that paper at the Society for Applied Microbiology in Edinburgh, UK in 2016. The paper is about to be published. So we also did some work on partial purification of cellulase. We presented the paper severally, and then we currently isolated some technological competent microorganisms from the abattoir effluent. So we want to use those organisms actually to see if we can use them for the treatment of effluent and wastewater. So those are part of what we have done. But I also want to discuss that there are other areas of waste to wealth that researchers can collaborate with us with. The char technology and compost infrastructures, for example, conversion of anaerobic digestate into activated carbon, compost infrastructures, especially where you have expanded food waste in the community. You can also use them for soil amendment, increase in soil microorganism population, because there are some other methods like applying NPK that are more dangerous to the environment. These chemicals actually kill the original microorganisms and worms in the soil. So by using waste as a soil amendment, you kind of rebuild the ecosystem. Then biodegradable bags can be produced from water hyacinth. We have a lot of water hyacinth in Lagos on the lagoon because of the additional nutrients. So we can use water hyacinth for the production of bio-oil, feeds, baskets, trays, and other decoration. We also have biodegradable bio-based plastics that can be produced using resins from green sources. which can be obtained from wood waste. Plant fibers for handmade papers, bags, and decoration can be obtained using using the waste from woods and from papers. Polylactic acids can be obtained from engineered microorganisms. Actually, even from the biochemicals that was produced by one of the microorganisms, we have a lot of lactic acid. These acids can be used for the production of plastics so that we will avoid chemicals that are persistent in the environment. Microorganisms can be used for various things, like in biobleaching, to increase the brightness of papers. We can also use them for detoxification of contaminated water and wastewater or sludge. We can use them for biomining, a situation where you can subject microorganisms to the waste so that the microorganisms will take up the metal. They are taking up the metal for their own growth as a substrate, but you can always recover the metals from microorganisms. As a result of that, you can reduce the demand for energy or the space of the landfills. Microbial technology for metal recovery from e-waste and solid materials from incineration residues is also possible. So in conclusion, researchers, scientists must gather the evidences to improve the manner that waste sustainability is valued. Shared scientific technological policy and community-based knowledge is required for transiting to sustainable waste and resource management practices. Policymakers must integrate environmental and social benefits and not just economic benefits alone. Most of the time, the policymakers are after the economic benefits, neglecting the social aspects. There must be interdisciplinary collaboration because there's no single entity that can handle the issue of circular economy. And focus must be on designing recyclability, reusability, and recoverability into products and processes. I said that earlier on. And opportunities will also be created for recovery of business, pilot innovation in existing industry, and expanding of employment and curtailing of poverty when we integrate circular economy into the system. Resource recovery and circulation and alleviation of risk associated with raw material importation and uneven distribution, especially in low-income countries. Just imagine if the COVID did not subside. Here in Nigeria, most of the time we depend on importation of chemicals. By now, people that would have gone hungry because most of the raw materials would have been depleted. So there is need to look into the wastes and how we can convert them to something useful. Resource circulation should be designed around eco-friendliness and renewable resources. So it shouldn't be born out of self-centeredness for making money. It should, first of all, consider eco-friendliness. That is what circular economy is all about. I want to say thank you for listening. We know we are open for collaboration. And I want to thank the following people that have made impact in my career journey. I want to start with Dr. Fred Mitchell, that freely gave me his lab in Ohio State University, US. I want to thank Dr. Sabri Griwal. She was of tremendous help during my program. I want to thank the Society for Applied Microbiology because they have been part of my success. I thank the University of Lagos because they've always given me the platform to be able to travel to do my research. I also want to thank Professor Dr. Christoph Sildak of KIT Germany because I was looking for space for my postdoc and he doesn't know me. I just wrote him and he accepted me and transferred me to Dr. Catherine Osentreta. That's a picture. So they are marvelous people. Of course, I need to thank my professors, my supervisors, Professor Aman, Professor Ilori, because they were the ones that actually spurred me to start this work, actually. And Professor Oti Ogundipe, our Vice Chancellor, who actually pushed me into circular economy. I thank Dr. Asma Ibrahim of Nasra, Abuja, Nigeria. She's a good mentor, she's a good mother. She was actually the one that encouraged me to make this presentation, because I was looking at it that what have I done that I want to present? I still need more time. She said, no, Maria, you have done a lot. Try to make the presentation. And through her encouragement, I was able to put something together. I thank Yisa, he's a good colleague. He's been very helpful. I thank my dear family. They've been very patient with me. My dear husband, I travel here and there. I stay late in the lab. He's been very patient with me. I thank my children, who has been very enduring. When I have to travel or be in the lab for days, they are there to support me. I want to thank Atlantic International Research for providing this platform. I thank you all, my listener today. God bless you.
Wow, what an excellent presentation, Dr. Maria Olanike Buraimoh. It has been a wonderful journey through your presentation, and the feedback we are getting from all over the world has been just one word: wonderful. Another word: great. Let me give you another one: excellent. So congratulations, Dr. Buraimoh, on this wonderful presentation. So we also appreciate our audience for the participation and the wonderful collaboration during the presentation. So we have so many questions here on the platform, and I would like to read all the questions. Dr. Buraimoh, maybe you can note them so you can answer them together, because some might be connected. Yeah, so we have Miss or Dr. Omnia Ibrahim from Egypt. So she has so many questions. And one of the questions is, in order to convert circular economy to, in order to convert circular economy, there is need for collaboration between researchers, research community, and all stakeholders. So, do you agree that how should there be an effective relationship between all stakeholders to have an effective conversion to circular economy? That's number one. And number two, she further asks other questions like what are the uses of algae bloom, and how can this be relevant for agricultural purposes and for making medicines for some diseases, especially maybe COVID-19 and the rest. And also, you have a question as well from Omnia that in some experiments, some plant residue of plants are used and used in circular economy, and some are as well used as feed for algae and for other nutrients. So could you please give us some hints about that? And we have Yaya Soro. Yaya Soro is from Ivory Coast. So Yaya Soro is telling us that what is the difference between the pelagic fish and the demersal fish in terms of exposure to bacterial infection. Now, which of the two groups is more exposed to bacterial infection? And does cooking help to protect infection from consumers? And so we have like some anonymous questions as well, that how long can it probably take to see excellent result or significant result on the well-being of Lagos Lagoon? For example, if there are support from the government. And do you know of any publication that has been able to analyze how cost-effective it is to apply your research to the government, and how they can be aware and how they can use it to improve the Nigerian economy in multiple industries? So we have other questions from Dr. Falilu Adekunbi from Neoma, from Lagos State. So Falilu is asking, now, what is the extent to which microbes influence climate? So moving from Africa now, going to Brazil. So Carlos from Brazil is asking, are these microbial strains common in other areas of the Atlantic Ocean, for example, Brazil, or each region has its different microbial biome? So Carlos further asks that, does the microbial biome varies seasonally or geographically within Nigeria? So we have Antonio from Angola. Could any of these microbes cause diseases? And last, we have Dr. Mark from France. Dr. Mark from France is asking, how can this process escalate to industrial level? And will it be easy to find enough litter to keep consistent production? And is it economically viable? So the last one from Marcio Silva de Souza. I think Silva de Souza is from Brazil as well, from Rio de Grande. So do you work with agricultural issues, or what do you know about this kind of research in Nigeria? Once again, Dr. Buraimoh, congratulations on your presentation. So you have the floor to give response to these questions.
All right. Thank you very much for your questions. I'm really impressed because a lot of questions shows that you were really paying rapt attention to the presentation. Nobody knows it all. I may not be able to provide answers to all your questions, but I will try as much as possible to answer the questions the best way I can. So the first question from Dr. Ibrahim from Egypt, need for collaboration between all stakeholders. Do you agree? Yes, I said it in my presentation. No one person can do it all. If you want to go into the work of circular economy, you need quite a lot of collaboration. All stakeholders must be involved. The scientists, the policymakers, I explained all that at the beginning, the policymakers, the NGOs, even the populace, those in the community, because if they do not cooperate with you, you cannot achieve anything. Let's look at plastic, for example. If we want to recycle plastic or we want to convert plastics, we need these citizenries to help in the collection of plastic. We cannot go to the street and start packing plastic by ourselves. But you know, to get plastic out of the street and invariably out of the ocean, what you can do is trade, a little trade. Tell the citizen, if you can bring so kg of plastic, you collect data for your phone to make calls, or you collect so amount of money, or there's so souvenir to be collected. So by so doing, they will all want to keep their plastic for exchange of something. So everybody is involved. Nobody is to be left out in this program called circular economy. Don't forget that our focus, my own focus, is to get rid of pollution from the environment and invariably from the ocean. So I hope that will answer that question. Alga bloom, you asked what it can be used for. Alga bloom is just excessive growth of algae, green algae on water as a result of addition of nutrients. And when you have sunlight, they begin to grow excessively. And I already told us about the implication. For example, it will not even allow the penetration of light. Oxygen will not be able to go into the water. It covers the surface of the water. And so imagine the organisms like fishes and other organisms below the water. So it is not something we want on top of the water. It can even affect the boats, the ships that are moving, navigating. So, but this algae and some other water plants have been known to be rich in some oil, and some of them can be fermented to be used as feeds for animals. So I hope that answers the question. The same thing with plant residues. Different plant residues, you have different plants. When you analyze plants, you have varied plants containing varied items. There are items that are common to all plants at various percentage. So it depends on which plant you are looking at. There's some grasses, some good plants are very rich in oil and nutrients, which can actually be converted to feed. But you know, I don't just convert that way. Most of the time, I inoculate microbial species. So in the process of the microbes taking up these residues, they also release some other substances as their own waste product or metabolic product, which will serve as a useful product to human. Yaya Zoro talked about pelagic fish. Well, I'm not a zoologist and I'm not a marine biologist, but I do know that fishes are affected by waste in the ocean. I talked about lignocellulose waste. When they get into the water, after a while, they get decomposed, and then you begin to see the components of woods, which I showed earlier, different type of polyaromatic compound and lignin-related compound that are very recalcitrant. So they will definitely affect the fish and affect the eaters. Now you talked about, does cooking remove bacteria? Cooking will remove bacteria when well cooked. It can remove bacteria, but mind you, some toxins cannot be removed by cooking. So if you have some toxins from polyaromatic compound or hydrocarbons in the ocean, or you have microplastic and their consistent toxins in the ocean, and it is absorbed by the fish, definitely it will affect the eater. It's not all toxins that can be broken by heat. While some can be broken by heat, others cannot. How long, how cost effective is, can it take Nigeria or say Lagos State? Well, Lagos State have been trying. Like I said at the beginning, I give it to Lagos State. They have gone a long way. Although it is highly populated, we have a lot of waste being generated. They have tried, they are still trying. But like I said earlier, Lagos State government cannot handle it alone. So if you have a network of collaborators, a network of scientists, and everybody is doing their own bit in their own place, and then come together with the result, I think there's going to be an improvement. There's already an improvement in Lagos State. There's an improvement, but there's still need to do more. I don't know if I answered your question. Are these microbes common in other regions? Well, I believe the microorganisms are everywhere, but the strains might be different from my experience. Don't forget that there are mutations. There are mutations. Other people have isolated Streptomyces silicola before me. They've also isolated Bacillus megatherium before me, but my own is Streptomyces silicola with some reference name, and then OB, Olanike Buraimoh. That is my own specific strain. And when you take another strain of that same Bacillus megatherium, which is not my strain, they may not be able to do the work that my own strain is doing. Remember that the environment will dictate how the microorganisms will adapt. Like I said at the beginning, these marine organisms are adapted to toxic environments because that is where they found themselves. So they've been able to adapt to cope with very harsh toxic environment. That is why when you put them in hydrocarbon, some of them will still survive. If I take the same strain from the US, it may not be able to survive the hydrocarbon because it has not been exposed for that purpose. Like, look, I showed you the diagram of some shanties where people live on the lagoon, eat, feed, defecate there, and they are healthy, they are okay. But if you pick another human being from another location, say from Ikoyi or from Lekki, and you take that person to that shanty, within 2 days, I'm sure the person is going to come down with diarrhea or some other infection, malaria. No, because so the same way with microorganisms, when they find themselves in an unsuitable environment, some will die, but the ones that eventually survive we have adaptation to survive in that environment, and so they become very powerful to do some other things. So I don't know if that answers your question, but collaboration means that we can always work together. For example, I actually went to Germany with my own stream and with my own waste products. So strains could be exchanged and all that, especially when you are sure that you are not working with pathogenic organisms. I'm not working with pathogenic organisms. So you have to be sure of what you are working with. I don't know if I was able to answer your question. Does microbial biome varies with season? Definitely. Sometimes season will affect microbial biome, and the activities that is taking place in a particular ecosystem can also affect the biome. When certain activities take, for example, if there's oil spillage, it will definitely affect. Some of the microorganisms will migrate, others will die, the survivors will mutate, and then change over time. Dr. Mark from France, how can this be translated into economy? All this can be translated. It's a question of cooperation between stakeholders coming together. If you look at several literatures, there are several literatures. Everything cannot be brought here in 40 minutes or 45 minutes presentation. If you look at several literatures, they've actually analyzed the quantity of waste that is produced or generated throughout the world, in each country of the world, and overall in the whole world. And they've been able to see that when you look at the quantity of waste, it can actually sustain this business. But don't forget that I am more particular about keeping the environment clean. So that is why I said the policymakers shouldn't just look at the economic benefits alone. They should also look at the social aspects to keep human beings safe, to keep the environment clean, because there are various diseases that are springing up as a result of climate change. And if we must maintain a good climatic scenario, then we must know how to keep or treat our waste. And the best thing is not to set them ablaze, so as not to release dangerous gases, the greenhouse gases into the environment, or not by setting them ablaze, so as not to release gases into our respiratory system, so as not to cause respiratory disorders. So the best way is to treat them and reuse them, even if it is for agricultural process. Nothing is really wasted. There's really nothing called waste. Nothing is wasted until it is wasted. That is where I'm going. Do you work with agriculture issues? I'm not an agriculture person, but because I do bioconversion, I have found myself honestly in almost all the fields, because the digestate from the anaerobic digestion, I cannot just throw it into the drainage or throw it into the water. What will I do with it? The first thing is to treat it and then use it as biofertilizer. And we actually carried out some experiments using NPK, the normal synthetic fertilizer, and using the digestate on some vegetable, and they produced, they gave us very good results. So meaning that even the spent is either I convert it into charcoal, or I convert it into biofertilizer, or I treat them to remove any potential dangerous microorganism before I spread them in the environment to allow the growth of the usual worms and microorganisms that are particular components of the environment. By the way, by the time microorganisms pass through anaerobic digestion, most of the pathogenic organisms are actually dead, because most of them cannot survive the environment of the anaerobic digestion. So I don't know if I've been able to answer those questions to your satisfaction.
Dr. Buraimoh, can you please share your screen again? Go to your last page where you have the collaborations. So thank you so much for the wonderful presentation. Particularly great thanks goes to the audience for their wonderful collaborations and attentive participation. Yeah, Dr. Buraimoh can be further contacted. You can see the contacts on the screen. So the email and the LinkedIn page. So for further discussion or further collaborations, because they are really doing a lot in their section and they are trying to see that some waste are reduced from the environment and the environment is clean through a circular loop and creating a green environment. And remember that the UN Decade of Action will be is on. So in the next 10 years, action is what is required. So we need to give more action and to work together to make impact, sustainable impact in our environment. So on this note, I would like to put an end to this Friday's event. So on this note, see you again next Friday. So thank you so much, everyone. Thank you. Bye-bye.
Bye, everyone.